Oh, that is close. I don't know about how close that is. I don't know how to zoom out. Um, that's okay. Well, it's fine. Cool. It's not as bad once it's on the little. We'll just hide that. There we go. One, two, three, four, five, six, seven, eight. I already lost count. Four, that's 8, 9, 10, 11. Okay, 11. Who do we have online? Anybody? No, anybody? Not yet. Make sure that we're signed in. We're gonna get going. We're not gonna do any of our Kahoots completely through, maybe Kahoot 10. Now, if we did our concept map, we probably realized chapter 10, pretty big boy, pretty big boy. Chapter 9 has got a lot in it, but a lot of it is pretty straightforward stuff. So we'll spend some time on 9. We'll go through 10 very quickly. We'll spend some time on 9, go through 25 very quickly, and then try to spend as much time as we can on 10, right? Especially clarifying what I want you to know from 10 versus maybe what I don't want you to know from 10. So we'll try to pace out our stuff like that. We will not make it through all of chapter 9 or 25's Kahoots. Both of them are 10-question Kahoots. And if you want to use them for study purposes, remember you can access them in your modules under the lecture application activity. There are links to those Kahoots for study purposes if you want to do that. Okay. Yes, it is. Good. So we're going to get going with chapter 9. Actually, before we get going. I lied. Did I close it? I did close it, didn't I? After all of that. Oh no, it's open. It's just in a weird tab. This is what I want. Okay. This thing that I have open here. This is what we have left until the middle of our semester, right? I wish I could just open this in a separate thing. There we go. Open file. Beautiful. There, that's much better. All right. This is today, 9:30, Tuesday. We're going to, in lecture, be going over chapters 9, 25, and 10. And in lab, we'll be doing lab 10. Lab 10 is mostly me walking through concepts. So remember how you guys did your pre-labs for lab 10? There were like 4 different ones. It was like transformation, gel electrophoresis, and PCR, DNA-based stuff. And so we're going to be doing DNA-based stuff review. We learned this stuff in chapter 8, but in the lab, we're going to really walk through the processes to make sure we're understanding those processes, because we need to know it for both the lab midterm and for chapter 8 as part of unit 2. Most of our lab today, and it's going to take a good portion of the lab. Again, I expect that for this class, it's probably going to take less time than I did for the other classes. So hopefully we'll get done a little bit early still, okay? We're going to shoot for that. The lecture today, because chapter 10 is so hefty, some of lecture might carry over into the lab time, but if we're as efficient as I expect we will be with the lab, we'll still probably finish overall early, okay? So that's the idea. Hopefully we get early in both of them, but I'm not making any promises, okay? But we won't go over time for sure. So anyways, we'll be doing a lot of blabbing and walking through, and I will pull up my tablet and I'll walk through it with drawings for everything so we can visualize it and see how it works. So that'll be a lot of lab 10 today. Thursday, obviously we don't have lecture class on Thursday. So there's nothing going on then. But in lab, we'll be doing lab 11. And lab 11 is the ELISA lab. It's going to be antibodies and antigens, which are brand new concepts for us. And we'll talk through that stuff in lab 11, and it will prep us for unit 3 coming up, because unit 3 is going to focus on antibodies and antigens quite a lot. Okay. All right. Next Tuesday, obviously in lecture, we will be doing chapters 13 and 14. The lab review will be on that day as well. So where's my pen? Here it is. For this day here, so next Tuesday, you can just join via Zoom that whole day if you want to. I will be in here in this classroom if you want to show up for chapter 13 and 14. You do have to join live. This is not a, you can watch the recording thing and get points situation, unless you have extenuating circumstances that we discussed, okay? Same situation there. If you want to join the Zoom via Zoom live, and then I call on you for your questions and you can type in the answer or verbally state it over Zoom, then that still counts as attending. Make sense? Because the lab review will also be given over Zoom. I will also be here in person in the lab doing the Zoom in there. So I'll move to the lab and Zoom whatever I'm reviewing in the lab. So if you're here in person, it'll be a normal day for you. But if you want to join via Zoom, you can Zoom the whole class that day if you want to. So that's your option there, if you want, okay? I will send out a reminder email on Friday to everybody about what link to use and reminding you that you can do Tuesday virtually if you want to, yes.
Physical things.
There might be some physical examples of some of the stuff, but most of it will just be pictures, because a lot of it is slides, and like me having a slide physically, it doesn't make any sense, right? So having pictures it is a Kahoot.
Okay.
So it'll have some pictures, and the most that I'll have physically might be like a Kirby Bauer plate, but like the pictures do justice for those. I don't feel like it's necessary. So most of that can be pictures. It will be on Kahoot. I will also share the link to the Kahoot, but again, you guys know the drill with the Kahoots. Me walking through it and breaking everything down is how we're going to be doing that. And I won't be calling on you for those Kahoots, but you guys will be participating, and I'll be going over, like, if we have a Kahoot question that's like, what's the primary stain for the gram stain? I'll be talking about how that primary stain works, why it works, why the gram positive is purple and the gram negative is red, and everything like that. We'll be breaking down those questions quite a bit. But you're welcome to join via Zoom, and it will be, I feel like, equally as productive if you're in person or virtual on that review day. Now, this will come up again when we get to our lab final, and our lab final review day is better in person because I have actual tubes to hold up, and it's just easier to actually see the tubes and be able to manipulate them. So when we get to that at the end of the semester, again, I'll remind you about that. Okay, but for now, I feel like the virtual is just as effective as the in-person for the midterm review. Okay, so that's next Tuesday. Next Thursday, Obviously, Thursdays, we don't have lecture anyway, but next Thursday is our lab midterm. So be aware of that. So that's next Thursday. The Tuesday after that, we have our Unit 2 exam that's going on that day. And we won't have any lab on that Tuesday. We never do on our unit exam days. And as it turns out, the Thursday after that, we don't have a lab either because it is fall break. So just FYI. We're tracking. So this is going to be quite an experience of we will have lab next Thursday and we won't have an actual lab again until after fall break. There's quite a break in what us actually doing wet lab work, interactive lab work, until we come back. And that is why when we get back, we're gonna get new tubes for our unknowns. It's the same bacteria, but I'm gonna give you new tubes of them because we used up all our old ones. And you're gonna do gram stains on them to re-identify them. And we're gonna make sure we're caught up on our unknown sheets on that day. That'll be our first day back after the fall break. And then moving forward, we should be very well set up to keep filling out those sheets as we go. So no worries if you haven't started them. When we get back from fall break, we will make sure everybody is on the same page with those worksheets. We good? Yes. Awesome. Okay. So that's what to expect. Just showing it here so that you have some reference for it. All righty. Let's go. All right, let's get going with our chapter 9. Actually, let me join online. But she wouldn't let me hear it. Okay, first question for chapter 9. The acronym CHOMPS is used to refer to the elements needed for life. What is S? Basil is not the thing. I don't want to clutter people. Okay. All right, so S is sulfur. S will be sulfur in that. So not even sulfate necessarily, sulfur. And that's going to be more important in our amino acids. Methionine and cysteine have sulfur in them, and they're capable of making disulfide bonds in our protein structure, which we will review again because I know it's chapter 2 stuff, but understanding protein structure, now that we're getting into talking about enzymes and denaturing them for chapter 10, we'll review that stuff because I feel like it gives, now we have a better perspective on why it's important and denaturation of those proteins, how they can be affected and stuff like that. So this gets to our first question. Tanya, you're going to be our first person. Yes, there you are. Okay, I was like, I know you're there somewhere. Okay, there you are. All right. We have C is carbon, H is hydrogen, O is oxygen. What is N? Nitrogen. Easy. I feel like these are pretty easy. I want to keep them kind of light because these are kind of going to get heavy chapters. All right. All right. Jonathan, what is P? Phosphorus. Excellent. And I like that you said phosphorus and you didn't get tripped up with phosphate because the P itself is phosphorus and the O's around the P are the oxygens. We're talking about the literal atoms here, the elements themselves. So we usually see phosphorus presented as phosphate. That is true, but the P itself is the phosphorus. I'm going to draw over here really quickly. Well, actually, let me hold off on it. Yeah, we'll hold off on it. Okay, so we said that our N was nitrogen, right? So I know that the nitrogen cycle was chapter 25, but we're talking about nitrogen. Let's go over the nitrogen cycle and what I expect you guys to know for the nitrogen cycle. Nitrogen cycle. All right, in our atmosphere, we have N2. In fact, most of our atmosphere is N2. What you're breathing in right now isn't pure oxygen, right? It's like 20% or something. I don't even know the exact percent. It's like 27% oxygen. Most of it is N2, nitrogen, that you're breathing in right now. And so that doesn't serve us very well. We can't use that nitrogen. What are some molecules that we might want to use nitrogen for? Nitrogen-containing molecules. Molecules. There's 2 major macromolecule groups that must have nitrogen in them. Evelyn, when we think about nitrogen-containing molecules and our macromolecule groups. So our macromolecule groups, just to review for everybody, we have 4 macromolecules: carbs, lipids, I don't want to give this away, nucleic acids, and proteins. Do you know which ones need nitrogen? You can name one of them if you want. So we'll start with the nucleic acids. Beautiful. Nucleic acids have to have nitrogen in them. Now, this is to remind you, what are nucleic acids made up of? Nucleotides, right? And so what is a nucleotide? You don't have to answer this. I'm just saying this. I'm going to tell you what it is, okay? So we know a nucleotide is a phosphate, PO4, linked to a 5-carbon sugar, either ribose or deoxyribose. If you're deoxyribose, we're talking about DNA. If you're ribose, we're talking about RNA, right? And then that is also linked to a nitrogenous base. We'll put NB just so to remind you, it's a base, but it is a nitrogenous base. It even says it in its name that it's got nitrogen in it. These nitrogenous bases are A, C, G, and T or U, right? Yes, so we know that's a nucleotide. Obviously, you would need to have, I think they can now see my screen. Hopefully they see my screen the whole time. Obviously, here we have nitrogenous bases have nitrogen in them there. The other one of our macromolecules that has to have nitrogen is proteins. Proteins are made up of amino acids. And it's in their name too, believe it or not, if you remember what an amine group is. So an amino acid, we have a central carbon that is connected to an NH2 group. I could put NH2, but I'm going to draw it out in the structure, okay? N with 2 H's. That's an NH2. And that group is known as an amine. Okay. That central carbon has a hydrogen on it because we got to keep 4 bonds and keep carbon happy. We have an R group attached to that central carbon, and we have a carboxylic acid on the other end. This is the acid part of amino acid. So we need nitrogen in our proteins and nitrogen in our nitrogenous bases. So without nitrogen, you don't have your genes, you can't transcribe them into RNA, and you can't translate them into protein. Obviously, we have oxygens in there, we have carbons in there too, but I'm just saying, don't forget that the nitrogens are equally as important as the CHs and Os. And the phosphates too, of course. Okay, so nitrogen, back to our nitrogen cycle. We know why it's important and why we need it. So nitrogen in our atmosphere is essentially useless to us. As it turns out, there are bacteria that can convert nitrogen in our atmosphere into ammonia, NH4+. I know this is an ammonium ion. I'm not concerned about you knowing the difference between ammonia, NH3, and ammonium ions. They're interchangeable for my purposes. Now, when we're converting nitrogen in the atmosphere to ammonia, this process is known as ammonification. Okay, that's all you need to know about it. I don't need you to balance an equation or anything like that. Taking nitrogen from the atmosphere, N2 gas, and turning it into ammonia, bacteria that do that are going through the processes of ammonification. We tracking? You also, if you eat food, and we already talked about how you eat DNA, that has nitrogen in it. We also eat proteins. And if this amine group is an NH2, you can imagine if you're breaking down proteins, that can easily make ammonia, NH3, and NH4 pluses as a byproduct. That's not ammonification, but it is something that we do, and we excrete ammonia as waste, and that'll get incorporated in soil as well, which is why we use fertilizer, because a fertilizer has ammonia in it, and bacteria can also make ammonia through ammonification. So these are just sources of ammonia. We can draw that on our little chart too. We got a cow. I don't know. This is our cow. Okay. He happy. And he pooping. Here's his poop. And then that also leads to ammonia. It's not ammonification, but it adds to ammonia in the nitrogen in our world. Okay. Back to our cycles. Some bacteria can convert ammonia into nitrite. Nitrite. And even further into NO3 nitrate. This whole entire process of going from ammonia to nitrate, it's a two-step thing. This process is called nitrification, also known as nitrogen fixation. Okay. Don't need you to balance the equation. Just know that converting those turns it into that. Involved in this, plants. Here's a plant. I could have just drawn a tree, I guess, but here's a plant growing in the ground, and here's its little roots. Plants use nitrate. And then we eat the plants and get our nitrogen from the plants. So plants use nitrate, but they can't just get nitrate from the environment. Bacteria have to basically convert it in these steps. Are we following? Okay, easy enough. Some bacteria also convert nitrate. So our bacteria are in the orange here, what they're using, and they're part of the cycle, okay? The reds are kind of our side steps and how macroorganisms are involved. Some bacteria convert nitrate into N2 gas again in the atmosphere. Somehow we've come full circle. This is a process called, anybody remember what this process is called? It's okay, denitrification. That's it, that's all three of them. Ammonification, nitrification, and denitrification. So you can think of N2 in our atmosphere is not part of biological organisms. Our bacteria convert it into ammonia, nitrite, or nitrate, which are part of biological organisms using it. So we either ammonify it and then start the process and then go through nitrification to turn it into nitrate where like plants and other organisms can use it. Denitrifying is removing it out of that biological system, removing the nitrogen, putting it back in the atmosphere. Okay, so those are the three steps. We following with the nitrogen cycle. That's all I need you to know about it. You don't need to draw it. You don't need to identify steps in it. I'm not going to have images on our exam of anything. They're all going to be word questions again. Okay, I know some people that think that sucks, and you're welcome to draw the cycles on your cheat sheets if you want to. I say cheat sheets, your scrap paper when you do your brain dumping. You get it. You're welcome to draw them if that helps you from recall. Any questions about the nitrogen cycle? Okay. Are we done copying this? Because I'm going to delete it now. Good. You say. Next question. Which of the following is heterotrophic? I feel like one of these things is not like the others. So hopefully that should help. I usually abbreviate energy, E with a not at the top of it. You might have seen this in chemistry. I don't know if they abbreviate it that way. I learned it from chemistry, but I don't know if you did. Anyways, so I don't have to keep writing energy, but When we talk about where nutrients and energy come from, we often define organisms as far as where they get their carbon from and where they get their actual energy from. And I don't know why I always write it out this way, because I always start with the energy, guys, because that helps me remember it better. And I always end up writing it second, but whatever. We're going to start with the energy, guys. If you get your energy from sunlight, we call you a phototroph. These are all trophs. Okay. You are a phototroph if you get your energy from the sunlight. Now, can you get carbon from the sunlight? No. Can sunlight help you fix your carbon? Yes, that's what photosynthesis is. But the sunlight doesn't directly give you the carbon. It gives us the energy to fix the carbon. Okay, so that's how you can separate. And that's why I always start with the sunlight, because this helps me keep track of the other three. Okay, I say the other three. Your textbook has more than three. It has more than four. It has a lot. But there are four major ones that I want you to know. Okay, so phototrophs get their energy from the sun. All right, if you get your energy from breaking chemical bonds, it's in the name, yes. I hear you saying it. I don't know who said it. Who said it? Okay, all right. All right, so I know. I think you say chemotroph. You said it under your breath. I heard it. She said chemotroph, and she is right. So I'm giving you points for that. Yeah. All right, so chemotrophs break chemical bonds to get energy. And we know that this is going to come up again in chapter 10, right? Because we know that there's a term in our overall system of how we're breaking bonds and making bonds with the metabolic reactions, right? If you're breaking something to get energy out of it, we call that anabolism. Okay, there it is, yes. I already called on you though, dang it. Okay, so catabolism or catabolism, either way you want to say it, as long as you can recognize it on a test. And we'll come to that when we get to chapter 10. But this relates to that, right? If you're breaking chemical bonds to release energy, then that is a chemotroph. Okay? All right, so it'll relate to that. All right, if you're getting, so those are our two for our energy, guys. For our carbon sources, if you're getting carbon from an inorganic source, we'll put a little I for inorganic, in the form of carbon dioxide. We'll put it in a parentheses to let us know that is our inorganic. This is where a lot of people get confused. If you took chemistry and somebody told you if it has carbon, it is an organic molecule, they failed you. If carbon is bound to hydrogen, then it is an organic molecule. So here's another example that I want to show you. Anybody know what this is? So it's called sodium bicarbonate. Okay, we're going down here. Okay, it's sodium bicarbonate, also known as baking soda. All right, the way that we draw it, and I'm not going to make you draw it on the example, I just want to show you something, okay? The sodium ionizes from the carbonate. And I think it's like, I'm not going to try to balance it, okay? But it's like this or something, okay? But there's two on this one, and we would have two of the bicarbs, all right? But this is going to ionize with one of the O's. We know carbon only makes 4 bonds, period. So carbon's going to go like this. It's got 3 oxygens. There's your H, and it is covalently bonded on there. Is that H bound to the carbon? So is this an organic molecule? No. Even though in this equation we have an H written near it, in the actual chemical structure of it, because the carbon isn't bound to the H. This is not an organic molecule. So we have to understand about the chemical structures in order to really understand what organic molecules are. So if we were to draw, methane is our simplest of all of our organic molecules, but as long as we have at least one H bound to a carbon, that dictates what an organic molecule is, okay? This is methane. So this is organic because we've got at least one carbon bound to a hydrogen. So that's why carbon dioxide is inorganic. But if you can convert carbon dioxide, which is inorganic, into an organic version, in this case, what do plants convert it into? 6H12O6. What does this represent? Beautiful glucose. Who said that? Ellen just killing it today. Okay, so glucose. It's a 6-carbon sugar. It's an organic molecule. We've got hydrogens coming off of carbons everywhere. Okay, so these organisms that can do this, we call these autotrophs. We've got phototrophs that use the sun. If you use the sun to make the energy to fix inorganic carbon into organic, you are a photoautotroph. We tracking? The energy is the sun, energy is from the sun, and we use that energy to fix inorganic carbon dioxide into organic glucose. Good. The other version that you can be is what this is talking about, heterotrophs. So they are using organic carbon sources like glucose or anything, organic carbon, and then repurposing that into other organic carbon. That is heterotroph. You don't have to draw it the way that I'm drawing it, but this is just letting me know I start with organic carbon and just convert it into other forms of organic carbon. Where do we get our organic carbon from? The plants, the photoautotrophs. We eat them, or we eat the animals that ate them, right? Okay, are we following with this a little bit? Because I feel like the next part kind of ties in a little bit with this. Yes, okay. I'm not gonna ask a question about this one. We're gonna go to the next question. I'm gonna leave this stuff up here for it. Next question, saprobes. What do saprobes do? Okay, so these are the guys that break down dead matter and detritus or detritus, however you want to pronounce that word. I don't care, as long as you can recognize it on the test. I say detritus, but you say it however you want. Detritus is fine also. It's like if you guys, anybody in here have Dr. Singh for chemistry? Anybody? He says, how does he say? I say capillaries. He says capillaries. That is weird. That's because he's from Trinidad. So a lot of people think he's from India. He's not. He's from Trinidad. And so that's how they pronounce it. They're the English people from England taught them how to speak English. And so they say capillaries. I have to think about it. It doesn't even come out of my mouth. So whatever, but stuff like that. It's the same kind of thing. Okay. Anyways, so yes, they decompose dead matter and detritus. If they decompose things that already had organic molecules, would they fall into any of these other categories that we have up here? Anybody want to volunteer, yes or no? Saprobes. Anybody know? So the saprobes would be heterotrophs. They're breaking down. They'd be chemoheterotrophs, just like us. We're chemoheterotrophs. We break down chemical molecules and we get our carbon from organic molecules from other organisms. Same thing with the saprobes. They're not using sunlight. They're not using inorganic stuff, they're breaking down existing organic molecules and they're breaking chemical bonds to get their energy. So we can think of saprobes as being a subset of the heterotrophs. Obviously, we are heterotrophs also, but we're not saprobes. We don't break down dead and decaying things. We might like to think of ourselves as humans as special because we kill the animals and cook them before we eat them, but they're not dead and decaying usually by the time that we eat them, right? And so we are just like any other predator or omnivore. The plants that we eat, they're freshly dead. You know, the animals that we eat, we preserve them so they will be as freshly dead as we can if you eat animals. You know, not judging there. Okay. Hope that makes sense. Let's move on to our next question. Not how much to say about saprobes, sorry. All right, diffusion of water molecules through cell membrane with the concentration gradient. What's the term? Okay. So this is called osmosis. Anytime that anytime water is what is diffusing, then that is osmosis. So we have diffusion. Diffusion is movement of molecules with the gradient. What I mean by that is moving from an area of high concentration to an area of low concentration. Do you guys remember when you took chemistry, you might have learned about this concept called entropy? You might vaguely remember it. And it might be this vague concept, but the idea, the law, it's a law of thermodynamics. And this law of thermodynamics, not a theory, it's a law. So this law of thermodynamics says that all of the molecules in the universe tend towards disorder. What that means is if I stack up a bunch of balls here, they want to fall and spread apart. We like to think, if we're being simple-minded, well, gravity is making that happen. But they will spread apart even in a vacuum of space, without any gravity at all. They'll want to eventually spread apart. That's entropy. It tends away from order. Okay, and that is a law of physics. That's not a law of chemistry. So this should help us put into mindset whenever we start talking about molecules. We start talking about water, for example. Well, that's two hydrogens and an oxygen. What is a hydrogen? It's a proton and a neutron with one electron circling it. What is an electron? Now we're in physics. So chemistry really is physics, and biology really is physics when we break it all down. So we don't really get away from it. So it's kind of a shame that we don't spend as much time taking physics because it really does explain literally everything, biological and non-biological, but it's very, very, very complex when we get it down to that level. But it's cool stuff. So anyways, diffusion physics says that if I have a whole pile of balls here and I let them go, they're going to spread out. That's diffusion. So physics says that is what's going to happen. And that physics says they want to be even everywhere, all over the place, and spread out all over the place. Just like when I spray perfume, that it's going to be strong here first and then diffuse out through the room eventually. And eventually, given enough time, it'll spread out completely, right? Whether it's in this room, seep through the cracks in the walls and whatever outside of this room, it'll diffuse that way. So we get the idea of diffusion. Diffusion is the spreading out of any molecule. And then diffusion of water molecules is osmosis. So we're tracking that, right? And the reason that the water is moving in this case is because the other can't, whatever your other is. So if I put a cell that has a selectively permeable membrane, and 95% of our cell is water, then that means that 5% of it is, we'll say other. Doesn't matter what, it's not water. Let's say we put this cell into a solution. And in that solution, the outside is 50% water. That means it is 50% other. Doesn't matter what the other is, yet again. If only water can cross the membrane, which way is the water going to go in this example? out of the cell. Water, and this arrow represents the water. Because the other can't move, the water does. So that's why osmosis is occurring, because it wants to equal out that water and the other, but the other can't move. So we're only moving the water. We're tracking that, yes. Okay, so that's what's going to happen. So this will cause the cell's shape to shrink, shrivel down. And that process of the cell shrinking, we call it plasmolysis. And it's driven by what we call osmotic pressure. It's why when we pack food in salt, it draws the moisture out of the cells. And it keeps the cells from being able to grow, because if they've shrunk like this, they can't live. It can potentially even kill some types of cells, right? So we know this about salt. But did you know if you pack the cells in sugar, it does the exact same thing? It doesn't matter what your other is, if all you can move is the water and it wants to equal out. So that's what jam and preserves are. That's why jam and preserves have so much sugar in them, because we're using the sugar to create osmotic pressure, causing plasmolysis, the same way that salt does. Salt is the more obvious example, but sugar works the exact same way. That's how jam works. Yeah? All right. But we have to have high enough sugar. Otherwise, they'll just break it down as a food source and it doesn't work, okay? So we have to have that concentration going. So we're following this, and this seems to make sense with us. If we were to, so that's our idea with a solution. Let me see if you guys can go further with this before we get to the questions about it. Jessica, the solution out here, what would you say that this solution is, the term for it, compared to the inside of the cell? So this is the tonic. Hypotonic. So this would be, outside solution would be hypertonic. It's okay, that's okay. So whenever we have a higher other, that's hyper. So the hyper and the hypo, that's okay, because we're thinking about that. I'm still going to give you credit because we're getting into the discussion of this, okay? So it's hyper. We know hyper means up. We know hypo. We know this, but this is in relation to the other. Is the other higher or lower? The other is higher in the outside. Now, if I were to ask you the cell's perspective, what is the inside of the cell compared to the solution that it's in? That would be hypo. We tracking? So what will always be given in perspective, always, if I ask you a question about that sort of situation. And you'll have questions like this on the test. It'll be a scenario. If the cell is 95% water, you only have room for 5% other. That's it. It's 95% water, 5% other. And I say that we're going to put you in a solution that is 50% water. You know, what is that solution that we're putting it in compared to the cell? So I'll always say, I'll ask you, is it, you know, the solution, hypertonic, hypotonic, whatever, or the cell, hypertonic, hypotonic, whatever. Now we'll make it clearer than I'm even wording it right now in the written form. So here, our solution is hypertonic to the cell. The cell is hypotonic to the solution. Are we okay? Is everybody tracking that? Okay. All right, that's good. We picked on you. All right. All right, so let's see if we can answer some questions about it. I'm going to leave this up and you can use it as reference if you want to. I think it asks us next. Okay, this is similar. What is the solute in salt water? So this relates to that. If our solution is NaCl and H2O. Which one is the solute? This is salt water. So the salt is the solute. CLO. CLO here today? Yeah. Okay. What would the water be? So if the salt is the solute, the water would be what in the solution? The other term. So solutions are always made of solute and the other term. So solvent. So yeah, I'll allow that. So I'll give you the points for that. Duncan, we'll come back with another question for you. I know that's a weird worded question, so we'll come back with another question later. But yes, so that would be the solvent. And you guys know this. I know you do, because water is the solvent of life. You know it. So if you can remember that, then you can always remember that water is the thing that other things dissolve in. So if it's always solvent and solute, I get them confused too. I can remember that water is the solvent of life, and then remember, okay, water is the solvent. The thing dissolved in water is the solute. I know it seems circular, but it works for me. Maybe it'll work for you. I don't know. Because I always have to start with solvent. I remember in organic chemistry, we used acetone a lot as a solvent. So it's things that we dissolved things into, but whatever works for you. I asked Dr. Tim if he had a better way of remembering it, and he didn't really. So for what it's worth, okay? Because you guys are not alone in remembering solute and solvent. Sometimes we just get lucky with it. Don't fret. Okay. Solute concentration outside of the cell is the same inside of the cell. What is that situation? So we talked about hypertonic and hypotonic. There's another one. If it's equal inside and outside. Okay, so that's isotonic. I'm going to ask one more question. We'll see if you can get this one, CLO. I think it's okay. It's okay. Okay, if I put a cell that is 95% water and 5% other, into a solution outside is 100% water. Which direction will the water move? So it will move inside. And this is why, because there's more water and there's stuff inside of the cell, the water wants to water down the stuff inside of the cell so that they equal on both sides. We can't move the other. This can't move, right? Only the water can. So outside says, imagine it like this. You got a cell with a bunch of salt in it. And now we put it in pure water. The salt cannot move. The water wants to water it down. It's the opposite situation of packing the cell in the water, right? If you pack a cell in water, we know sucks the water out. If you pack salt in water, water's going to suck in. Make sense? Okay, so it's going to go in. That would be a hypotonic situation. Now imagine that this is fine. Now imagine that you have a patient, and we usually use 0.9% sodium chloride for IVs, right? My brain isn't doing things. We usually use 0.9% sodium chloride. Now, if we talk about what your cells are actually made of and what's in your cells, talking about the salt in your cells and equaling that out, 0.9% should be isotonic for a healthy, hydrated person. Okay, so we want to keep it isotonic because we want to encourage their cells to want to be isotonic to that 0.9% saline concentration. Okay, I use 95 and 5 because it's easier to see than me doing the math of like 99.1 and 0.9. Let's not mess with it. Okay, so but it's 0.9% in reality. That should be isotonic, and that means equal on both sides, just like this question was asking, right? Okay, if I gave somebody an IV solution of pure water, what would it do? The pure water IV solution would cause their cells to take up water, and they would swell and even burst, like Jillian's doing this. I see it. I know what it means. Okay. So it even caused the cells to burst. If I give you a little injection of pure water, your kidneys can probably accommodate that. If I give you a whole liter of pure water, your kidneys are going to have a hard time balancing that out with salt from the tissues and putting it back into your blood to make up for that in time to save your red blood cells from bursting. Once your red blood cells start bursting, your kidneys are going to try to help filter out the damaged material from that. And your spleen is going to try to filter out the damaged red blood cells themselves. And your body's going to be signaling, what the heck, we've got burst red blood cells. And that's going to lead to shock. We're going to have a decrease in blood pressure. We're going to have reduced blood flow to the brain because we're trying to maintain that heartbeat pumping blood to vital organs. If that continues for long enough, that can lead to death. Right? The same exact thing happens whenever we have incorrect blood transfusions happening. When the body attacks the wrong blood type, it causes this to attack the red blood cells and then damages those, and your body goes into a shock response and it can be deadly. We'll learn about that in Unit 3, but it's the same exact thing that'll happen if you burst your red blood cells this way. Now, would it be easy to kill somebody with pure water? No, it would take a lot of water for this to happen, to actually kill a person. Like if you literally, for somehow, I don't know how you did it, but you got pure water that you were giving in an IV to somebody, that could kill them. But likely, where are you getting it from? I don't know, right? Just saying, I don't know where you're getting that from. They don't just carry that in hospitals, right? In an IV bag like that. We usually have isotonic saline. This is also why when you drink water, we don't drink pure water. In fact, you can't really hydrate with pure water because it's going to be hard for your cells to be isotonic and then your gut and everything like that to deal with it. Yes. Yeah, so distilled water, 90% of the time, so we don't recommend, you don't even see it, like you talk to your doctor or whatever, they're like, we don't recommend that you drink distilled water. Yeah, you're not supposed to drink distilled water. Now, it's not going to kill you. It's just not going to hydrate you as well because you need electrolytes and minerals in your water. So we'll talk a little bit about water processing in chapter 25, right? So this gets us into that idea of why it's important that we add back minerals into pure water. We'll come back to this, I promise, because we'll talk about water processing, okay? But it's the same idea, the tonicity of that. So we're doing good. You guys are doing great. I'm going to clear these drawings. I don't remember if there's anything else good in this that I want to talk about. Facilitated diffusion. What do we need for facilitated diffusion? This is a little bit trickier of a question. So diffusion will happen naturally on its own if there's no barrier in between. But our membrane is a barrier, and our other water can cross the membrane, but other molecules typically can't. Okay, and so in order to allow that doesn't work, right? How do we get stuff in and out of a cell then? Things like food and nutrients and stuff like that. So we need to have things in place like facilitated diffusion or active transport. In facilitated diffusion, we're letting molecules move with that same, why did I put a B there? We're letting things move with the gradient still, but it's with the help of a channel or a carrier protein. It's still with the gradient, still going from high to low. So the molecules themselves will move themselves, but we'll just need some other way. It can't go through the membrane, so we'll give them another pathway to go through the membrane, okay? That can be a carrier protein, or it can just be an open channel, but typically it's gonna be a channel that we can close and open at our need, okay? So that's facilitated diffusion. The other one, active transport, is what it sounds like. It's very active, and if you're active, you need energy. What do we use as our source of energy for that? You love it. I know you do. ATP. All right. So we'll need energy in the form of ATP to help with active transport. This is usually either against the gradient, meaning we're trying to pile the balls back up into the pile. That's against the gradient. If you're eating food, for example, you might already have a lot of food in your cell, and there's just a little bit outside of the cell. So we're going against the gradient, but we want more of it in the cell. We'll use energy to gather it up. So some examples of active transport would be phagocytosis, as well as endocytosis. Both of these things bring things into the cell. Phagocytosis is just a type of, they're very similar. Phagocytosis is like eating up big things. Endocytosis is like, I have the key to get in, please let me in. And the cell says, yes, please come in, and lets it come in a very organized fashion. If we're going out of the cell, this is called exocytosis. All of this is active transport. All of it needs energy. This might be going with the gradient sometimes. It's either against the gradient or with it at faster rate. What I mean is, instead of letting the perfume diffuse throughout the room gradually, using a fan to help it, that would be using energy to help it go faster with the diffusion. Make sense? So you want to absorb in the nutrients outside of the cell, but you want it to happen faster than nature would let it happen. So we're going to use energy to force it. So that is how active transport works. Any questions about that? I don't want to go through the rest of all of the questions for this chapter, but this chapter also covered a couple of things that we didn't get through with our questions. The first thing was symbiosis. Okay, so in symbiosis, you have two organisms involved. At least one of those organisms must have that interaction happen for it to exist. This doesn't mean that these organisms are always involved in the interaction, but it means that that interaction must happen for it to live. What I'm going to say is obvious examples to start with, okay? So, yes, we can try to go over that a little bit. I feel like we'll go over it a little bit, yes. Okay. We'll get to that one next. Okay. With symbiosis, sorry, that threw me off a little bit, me and my ADHD. There are three types of symbiosis. They're not all good. Symbiosis is not always a good thing. Not always. All right, so we're going to be that clear. My first example is a tick, right? What type of symbiotic relationship did ticks have with us? Anybody know? What was that? Parasitism. Okay, good, good, good. Okay, we heard it. We got parasite. Easy one. When the parasite benefits, and the host is harmed. All right. And there's one that's kind of in the middle, commensal. And I don't need you to know the commensal organism because I feel like that one trips people up. So really all I need you to know about commensalism is that one of them benefits. The other one is neither harmed nor benefits. An example of commensalism, sloths. So sloths have algae on their back. The sloths don't really care that there's algae on their back, but the sloths climb up into the tree and bring the algae close to the sunlight. The algae is benefiting from that. In fact, it kind of needs that. It needs the sloth to bring it to the sunlight. The sloth doesn't care. Now, I guess we could say that maybe it benefits from camouflage or something like that if you're stretching. But generally speaking, is it really getting true benefit from it? I don't know that it is. Okay, so that could be seen as one thing is benefiting, the other one just doesn't care that this is happening at all. Well, you see how ticks as parasites. Are ticks going to survive if they don't have a host? No, they have to bite something and feed on its blood. They can't live without that. It's not like your doggy. I love my dogs too, guys, but my dogs used to live on the street before I owned them. They were fine. They lived without me. That's not a symbiotic relationship. That doesn't depend on that relationship, right? So symbiosis, one of them must be depending on that relationship. All right. The next one is another obvious one, mutualism. Both benefit. Example of this, your gut bacteria. Your gut bacteria get a nice, happy, safe home, and they get nutrients provided to them in that nice, happy, safe home. While they're doing that, they break down those nutrients, and their waste we can use as nutrients. They also help train our immune system on what's friendly and what's not friendly, and other things like that. So we're both benefiting, the bacteria and us. So that is mutualism. That's what you typically think of when you think of symbiosis. But symbiosis can be any of these three things. Okay, are we tracking with that? Okay. Let me see. Another thing that we talked about in this chapter was the growth curve. And we'll get to, I promise, Maria, we'll get to that other thing in a second. The reactive oxygen species. In the bacterial growth curve, we start with the lag phase. This is pretty straightforward. I just want to cover it and be sure that it's covered. And this is the exponential, also known as the log. By log, we mean logarithmic, okay? And then this top part is known as the stationary phase. It's stationary. It ain't going up or down. And then the last part is the death phase. This is, I just want to cover it so we see, number one, you have to know it for unit 2. But this is going to come back in unit 3 when we start talking about how disease, not even through chapter 13, our next chapter in unit 2. Okay. And when we start talking about how diseases present themselves. Have you noticed how, let's say I have COVID and I'm coughing a lot and I come near Jessica and I start coughing near her. Does she get sick immediately? No, it's going to be a couple days. She'll be fine for a couple days. And then she'll wake up with a tickle in her throat and she'll feel tired. Okay. That period of time in between when I coughed on her and she starts getting that tickle in her throat is known as the incubation period, which correlates with the lag. This is the organism becoming accustomed to its environment and getting just initially established there. Then in that first tickle of the throat, we are somewhere in here, that first sign or symptom. And those are different, and you'll learn that in chapter 13, I promise. The first sign or symptom, we call this the prodrome. This is when that population is starting to get high enough that your body is reacting with it, and it is reacting with your body. We all know the prodrome. You know what it is. You've been there. You have a situation where you're like, I know I'm going to get sick. The tickle in your throat, or you're tired, or you get a little bit sore in the morning, that's the prodrome. Then when you are full-blown sick, we are somewhere in here, in this area. This is what we would call the acute phase of the illness. Correlates in here. When you're at that peak, and you're even in that stationary phase, where you're at your max of your population of whatever organism is in your body, you're in your acute phase. Then when you start feeling better, when you're going through this death phase in here, we call this convalescence. Again, this is chapter 13 stuff, but it directly aligns with this growth curve. Okay. So we'll come back to it, I'm sure, whenever we get to that. Are we good with this stuff? Can I erase this stuff? I'm trying. We're a little bit over time from where I wanted to be to start chapter 10. Okay. The last thing that Maria asked about were our reactive oxygen species. In aerobic respiration, we haven't gotten to it yet, but we're going to in a minute. We go through glycolysis and Krebs and the electron transport chain. If you are in aerobic, what is our final acceptor in the electron transport chain? Oxygen. We usually do this. We take some H pluses and we take some electrons and we bring them together with a half of a O2. Sometimes this process is imperfect, and we make oxygens that have too many electrons or too few electrons, or a lot of different combinations of oxygens that are imperfect. Okay, it doesn't always lead to H2O like it's supposed to. Sometimes we will have H2O2. Sometimes we'll have oxygens with extra electrons and whatever, right? This oxygen with extra electrons we call superoxide. This H2O2, where we have an extra oxygen tacked onto H2O, we call this hydrogen peroxide. Our body has to be able to deal with these. And to take our superoxide and get rid of it, we convert it into hydrogen peroxide, which is still dangerous. These reactive oxygen species can still interact with molecules in your body and cause damage. But the enzyme that converts superoxide into hydrogen peroxide is known as superoxide dismutase. Then our body can convert hydrogen peroxide, which is H2O2, into H2O, which is just water, and oxygen. I'm not going to balance the equation. Please don't ask me to, okay? It's fly. But that's what it does, okay? And this is why when you put hydrogen peroxide on wounds, it bubbles, because it's making gas in the form of oxygen, okay? The enzyme that does this is called catalase. That's the extent of what I need you to know about reactive oxygen species and how our body deals with them. If you are an aerobic organism, you risk making these guys, right? You got to have a way to defend against them. If you're an organism that lives in oxygen, then you need to have a way to defend against those guys. Does that help, Maria? I hope that that answered your questions, unless you have a specific question about it.
No, yes, I was just wondering like what it is and like what it's supposed to like pertain to, because it got me confused.
Yep, that's it. So just these ideas, dealing with these byproducts of aerobic respiration and being in oxygen, the dangers are superoxide dismutase, or superoxide and hydrogen peroxide, and dealing with those. That's it.
Okay, so it's the product of using oxygen as
Typically they're products of just dealing with oxygen in general.
Okay, thank you.
Yeah. All right. Cool. So we're going to be done here. Now. Clear. So it's going to give us our little podium. So we get a little bit of joy before we get into chapter 10. We're going to need it. Okay. So before we get into chapter 10, chapter 25, we already talked about our nitrogen cycle, right? There's the carbon cycle. I feel like that one's pretty straightforward. I'm not going to cover it. Okay. We also talked about this plastosphere and the effects of plastic in our environment. We talked about One Health, which is the idea we're not just worried about people being healthy, but our environment being healthy. And we talked about there was another thing and I can't remember what it was. Water treatment. Okay, so back to the water treatment thing. We know that we treat tap water that you're going to drink from freshwater sources, usually aquifers and rivers and lakes. For example, in Oklahoma City, most of our water comes from rivers and potentially aquifers, but they get cycled. And you guys probably don't even realize this. They get cycled through Lake Overholser, and that feeds into Lake Hefner. They're both important. They're both sedimentary lakes. They are literally there to let the water settle and let the junk in that water settle out. And then it flows into the next sedimentation lake, Lake Hefner, where it'll settle again. And then we can take that cleaner top water off and then treat it in order for you to drink it. So we'll go through all the treatment processes like you read about when you were setting up. Know the major steps in that, right? We're going to remove any solid debris and then any larger particulates through filtration processes, right? And then we're going to treat with chemicals and UV to kill any microbes that might be in there before you drink that water. Whenever you poop or pee and you flush it down the toilet, that goes through a different process of treatment known as wastewater treatment. In wastewater treatment, we are going to be filtering out all of the solids first. That includes your poopies, okay? solid material first. Anything that is solid that can be biodegraded, we call that sludge. Sludge can be biodegraded, and then we can take the liquid out of the biodegraded stuff and refilter it back in with the water stuff. Then we will go through the same exact filtration processes and even potentially treat it with UV and chemicals as well. And then release that wastewater that we treated, not into your drinking supply because it's still not as clean as drinking water, or release back into the environment. So this will be water that we use for parks, watering golf courses, as well as releasing back into rivers and lakes and streams and whatever. Might we also see that water again later on whenever we come back through the drinking process? Yes, we might, but we will refilter it even further before it comes back into drinking. We don't just recycle it over and over again. We recombine it with the environment and let the environment recuperate from it, and then we might pull from the environment again. So that's how water treatment is going to work. Now, the water that we treated here in Oklahoma City, does it just go straight into your mouth? No, it travels through pipes, through miles of pipes. And when is the last time you think those pipes were cleaned? I have RO water at my house. And now y'all all gonna be thinking about how clean your tap water is now. Okay? It makes me thirsty thinking about it, and I feel nice and safe drinking my water from my house. I live in Bethany. Bethany has some of the worst water in the entire state of Oklahoma, some of the worst water in the entire nation. It has horrible water. We're talking Flint, Michigan level terrible water. Okay. Bethany's water is horrible. It barely passes safety standards. And the only reason it does is because it has as much chlorine in it as they can legally put in it. It is ultra high levels of chlorine, and it is dirty water. They purchase their water from Oklahoma City and then also mix it in with their own water. Sometimes if they don't get enough water from Oklahoma City and they're using just their own, we will have boil alerts in Bethany. And it happens probably every couple of years. If you lived in Bethany or know somebody that lives in Bethany, you've definitely heard this. E. coli risk in their water supply because they are not properly treating their water. If you have well water, you're still at high risk for this. People who have well water don't realize you're just pulling bacteria out of the soil. Nobody is testing that. I hope you are. Did you have a question?
No, I was gonna say, I've lived in Piedmont like my whole life, and we've always been based on well water, and we've had similar things.
You can get people, I don't know what it is. Some people are like, oh, it's well water, it's clean. What are you talking about? It's not treated. It's just water from the ground. Oh,
It's like white.
yeah. So bathing with that stuff is typically fine. Okay. But if you're drinking it, it's a different story. If you're cooking with it or something like that, boil it before you're dealing with that. Right. And so when we get that, it's boil alerts that we'll get in Bethany. They'll be like, you need to boil your water. I have a reverse osmosis system at my house in Bethany. It wasn't cheap getting it installed, but I can tell you, you can taste and smell the difference in the water. My water that comes out of my tap, it smells nothing at all. You smell the tap out of normal Bethany water, it smells like chlorine. You can tell the difference. And when you wash your hands with the water, it takes a lot more soap to suds in Bethany water, whereas RO water, tiny bit of soap, and I'm getting full suds all day long because there's just so much junk in that water. It's not clean water. So you can feel the difference and you know the difference. But reverse osmosis purifies out the water, but there's no minerals in it at all. That is why in reverse osmosis systems, you also have to add what we call water softeners. So it's adding the minerals, remineralizing the water so that I can drink in it and bathe in it and my cells are still happy and whatever. Turns out that my pipes in my house are also very old. My house was built in the early 60s and they are, they're not PVC pipes. They are, cast iron or whatever, and they are corroding. So if I just have RO water and I'm just running it back to my house, I'm getting corroded pipe material in my tap water. So I have a spigot at my sink by my fridge that is specifically refiltered again. It's got filter and UV on it. And so my tap water is remineralized and refiltered and is nice and clean and happy. Okay. So I'm not saying your water isn't safe. I still drink from water fountains and stuff like that. But if you start noticing a difference in your skin health or your inner body health and stuff like that, you might think about, drinking bottled water, mineralized mineral water, right? Normal water that has remineralized in it, just saying. But generally speaking, Oklahoma City in general is fine, okay? But if you live in Bethany long term, that can cause some issues, and it has caused long-term. They had leakage of waste from Wiley Post Airport. It's been proven that the waste that they were storing at Wiley Post was leaking into the ground and into the water supply of Bethany for a while. That's Bethany. I mean, I like living by the lake, but it's Bethany, man. And it's behind the times. Okay, so moving on. We're not going to play chapter 25. We're going to move into chapter 10. We'll go over briefly our little activities. They are very straightforward for these. What is the difference between diffusion and osmosis? Osmosis is diffusion, right? Diffusion is movement of any molecules with a gradient from high to low concentration. Osmosis is diffusion of water specifically. Saline is administered. We already did all this. Saline is isotonic to our patient cells, and if you give them pure water, the cells are going to swell up and burst. We did this. We actually talked about this. I just forgot that that's the thing. All right. COVID. Whenever you poop and pee and you have COVID, you're pooping out and peeing out COVID virus. We often think of COVID as being a respiratory disease. You contract it through respiration, but it actually infects any cells in your body, all of them. So it's not really a respiratory disease. It can infect literally your entire body, any system at all. And so you can poop and pee out the COVID virus in pretty decent quantities. So whenever we're filtering out the wastewater, releasing it back into the environment, and then filtering it back again so you can drink it, viruses were making it through the filtration process. Our filtration process is set up for us to identify bacteria. We call those coliforms, fecal coliforms. They are identifier indicator bacteria. What that means is if I take water out of your tap and I see normal gut E. coli and Enterobacter or Klebsiella, all these bacteria that are just normal in your poop, if they make it through the filtration process, and that means the really bad E. coli, Shigella, cholera, all of those guys can make it through the filtration process too. So if the indicator bacteria, normal bacteria make it through, the pathogens could make it through too. And that's what coliforms tell us. But do coliforms tell us anything about viruses? No, viruses are much smaller than bacteria. So just because you're filtering out the bacteria, the viruses could still be there. We had to worry about this back in the day of polio. Polio is one of the tiniest viruses that we know of, and polio only exists in people, and it's transmitted through fecal oral route. So people were drinking water that was contaminated with the virus. So we had to redesign how we were filtering our water systems back then. And so we had to reincorporate that for COVID because it's another virus that was found in our water supply. Okay? Same idea. You'll notice that we don't have polio here in the United States anymore. It was eradicated thanks to vaccination. Okay? You know, it's a very debilitating disease. We've seen images of this. We'll learn more about it in Unit 4. We'll talk in more detail about it. Polio does still exist in Afghanistan and Pakistan. I have a student who's from Pakistan, and she's from a rural area in Pakistan, and she says there are people there that actually do have polio. They do administer the vaccine in those areas, but just they have a very poor view of vaccination, sort of like the way that our country is starting to go a little bit now, just saying. And so right now, the risk of somebody, we don't vaccinate or treat our water for polio anymore here in the United States because we don't have it in the United States. And people don't just walk around with polio. You have polio, you suffer it. So those people aren't just traveling willingly into the US and spreading polio and getting us sick anymore. So we would catch that in customs, for example, if they were traveling like that, if they could even travel. Many of the people with polio are paralyzed to some degree, who probably wouldn't be traveling. Not to mention the people that have polio are living in rural Afghanistan. What is the likelihood that one of those people are going to travel to the United States and bring polio here? I mean, it's nothing. It's not going to happen. So we don't worry about it here. And it's not high risk for bioterrorism because it's transmitted fecal orally and just like disseminate it through the air or something like that, right? So it's just not a risk. But anyways, okay, so we're going to move on to chapter 10. We ready? I hope we are. Make sure you're signed in. Yes. It's also why they don't allow motorized boats on the lake. Yeah, it just, They don't allow motorized boats in the lake because they don't want you turning up the sediment. Yeah, they need it to be sedimenting. They don't want people swimming in it because we don't want your extra nasty bacteria in there. And we don't want boats, you know, scooting around in it because we don't want that happening either, right? They actually want to keep it as clean as possible. It is pretty nasty.
We're the nasty
With all the birds that live in it, yeah. But the likelihood of people bacteria contaminating it, they want to minimize that. I mean, it happens still, you know, you can still like windsurf and you might fall in the water. It's not like illegal, but they want to try to minimize that as much as possible. I mean, see what happens whenever we put people in chlorinated swimming pools. We put people in chlorinated swimming pools. How many of those people pee and poop in those pools? It happens all the time. It's wild. We'll learn more about the diseases associated with that in Unit 4. It's coming. Okay. All right, so we're signed in. We know what's coming. We're going to start off with the enzymes. All right, which term refers to all the chemical reactions that occur within the cell? Cassie, you're going to be my next person. All right, if metabolism is all of the chemical reactions in the cell, whenever we break down things, what would that one be, Cassie? Okay, catabolism, yes, catabolism or catabolism. I say catabolism. Catabolism is fine as well. However you want to say it, as long as you can recognize it on the text, yes. So catabolism. Cats destroy. I know I say it in the lecture, if you watch the lecture video, okay, I know I say it, but I mean they do, okay? Cats destroy things. They break things down like your sofa and the things that you love dearly in your house. Cats break things down. And when we break things down, this releases that energy. Okay? Catabolism. And so, easy enough. And I know this is going to be the next question in the Kahoot, but then when we're building molecules, this is known as anabolism. Think of anabolic steroids. So people take anabolic steroids to build muscle. This uses energy, right? If we break down our carbohydrates and make ATP through glycolysis, Krebs, and electron transport chain, and then use that ATP to drive DNA polymerase to replicate our DNA to build new cells, RNA polymerase to transcribe the DNA into messenger RNA, or even our ribosomes and tRNA. We're using that energy to make new cells. And that would be an example of using that catabolism breakdown to make new things, pairing those things to please go away. I hate when it does this. That is amphibolism. Tying them together. And you can use intermediates of any of these steps as building blocks for some of the other guys. That makes sense? Okay. Next question is very directly linked to this. What is anabolism? So Sonia's not here, right? I don't know if she's online. I don't know if we have online. We just have Maria, that's right. Okay. Yes, so anabolism, building molecules by using that energy. Okay. And this is just one of those times to be sure we're reading our choices clearly and we're understanding what the choices are there. Does anybody have any questions about this one? Before we move on, because I'm gonna go. I'm trying to click through these. Enzymes are an example of what type of macromolecule? Enzymes are proteins. Do we have enzymes that are other kinds of macromolecules? Yes, we do. But the only ones we'll be talking about in my course are the proteins. Enzymes typically end in the suffix ase. DNA polymerase, DNA ligase, RNA polymerase. And they usually also tell you a little bit about what they do. Not always, but often, okay? ATP synthase, for example, okay? All right, so enzymes. There's two major groups of enzymes. I know you're thinking of the 6 different types of enzymes. I'm not talking about that yet, okay? The 2 major types of enzymes I'm talking about are simple enzymes and conjugated. Conjugated enzymes are complicated enzymes. All right, so our simple enzymes are just made of protein. They might be made up of several polypeptides if pink is one string of amino acids and purple is another string of amino acids, but they still have to come together to work. It's still just protein only. In fact, if we have to bring our pink together with our purple, that would be an example of the quaternary level of our protein structure. Remember, primary is just our amino acid sequence. Amino acid, linked to amino acid, linked to amino acid, whatever. Right? That's our primary. Secondary protein structure is whenever those amino acids interact with one another to create an alpha helix or a beta pleated sheet. The beta sheet looks like this. We'll have a straightaway, then it'll fold back on itself. We'll have another straightaway. And usually what's happening here is these amino acids are interacting with amino acids on the other side. And our alpha helix will have an amino acid here and one here maybe, and even around the curves or whatever. And how these amino acids are interacting dictates whether you're going to create these things. So these are all dictated, those amino acids. Remember what an amino acid looks like, right? We had that carbon with the hydrogen. We had our carboxylic acid. We had our NH2 group. And we have an R group. That R group can be hydrophobic, it could be polar, or it can have an actual ionic charge to it. Or it can have sulfurs. Sulfurs only come into play with our tertiary level, okay? But if these amino acids are creating hydrogen bonds or ionic interactions, for example, stuff like that, this is all dictated by the R groups that are in their neighbors. Neighbor R's. That's what our secondary is, okay? So we'll put little lines between these so we can see them. We're tracking? Tertiary. This is where you might have an alpha helix and then a straightaway and then another alpha helix that wants to come across it and whatever. A whole bunch of different shapes interacting with one another, folding up on one another. And maybe something on the inside in here. Some interaction here might be hydrogen bonds. Some interaction down in here might be ionic bonds. I don't know what went wrong there, but ionic. All right, or maybe we have a covalent bond here between 2 sulfurs. This is called a disulfide bond. So now we're not just the neighboring R groups, but R groups across the structure are interacting. Right? That's with one polypeptide. A polypeptide is just a string, a straight single string of amino acids. We're tracking? The quaternary level is like what we see over here. we have multiple polypeptides interacting with one another. Okay. So if you are just that protein makeup at that quaternary level, then you are a simple enzyme. If you are that protein, and you need some other thing bound inside of you to work, and you are a conjugated enzyme. This thing, the green thing, is called a cofactor. If you are an inorganic molecule, an example might be magnesium, like we need in DNA polymerase. It's inorganic molecule, right? It's not carbon hydrogen based. Then that's an inorganic. We still call that a cofactor. Just plain. If you are an organic molecule, for example, certain vitamins, like vitamin B12, for example, we've got molecules that have carbons, and at least one of the things bound to the carbon is a hydrogen. Then those are called coenzymes. I hate to break it to you, but you already know one. Obviously, vitamins and stuff like that, but there's one that we have in our list of things that we have to know, and that is acetyl coenzyme A. Acetyl CoA. The co stands for coenzyme. It's a coenzyme, right? So are NAD and FAD, acetyl CoA. We need to know all of these, don't we? They should sound familiar. Okay. So those are coenzymes. Are we tracking that a little bit? Okay. Do you guys remember? Actually, I'm gonna ask a question. I can't just talk all the whole time. Dylan. What's the protein portion of a conjugated enzyme called? So if the whole thing, the whole functional unit is called a holoenzyme. This is the enzyme. This is the protein part plus the cofactor. What is the protein part called? I hope you got it starts with an A. Oh, we got some hands on Kaylee. There it is. Okay, apo or apo enzyme. Beautiful. That's okay. We'll come back to you, Dylan. You'll get another chance. You guys remember this? So we've got the holoenzyme is the apoenzyme, which is just the protein part, plus the non-protein part. If the non-protein part is inorganic, we just call it a cofactor. If it is organic, we call it a coenzyme. A coenzyme is a type of cofactor. It's an organic cofactor. Are we tracking that a little bit? Does that help out a little bit with that? I know the lecture is very linear, but I feel like drawing it out in a chart like this helps us see the relationships a little bit better, okay? All right, so those are our enzymes. Now our enzymes can be regulated. If we have enzymes and we want to regulate them, we often use inhibitors. With our enzyme regulators, I'm going to switch to a more visible color here. We can inhibit in two different places on the enzyme. If this is your enzyme, this is your enzyme, it binds to and acts on this thing. Dylan, what's that thing called? What do enzymes act on? So the thing that the enzyme changes or works on. So that would be, I'll help you out a little bit. It starts with the S. Stop me when you remember the word. There it is. Yes, you knew it. Do you remember the site where it binds in? The something site. I know, Maria. Yes. Okay. So this is the active site. I know you do. I know you do. So now we're just putting this back into context, right? So that's the active site. So enzymes act on substrates. They bind the substrate at the active site. If you want to block this, there's two different ways. If you have an inhibitor that binds at the active site, we'll put him in red. He mimics the substrate. Usually we'll draw some little guys coming off of him so you know he's different than the substrate, and he's blocking our substrate from being able to bind in. Now our substrate can't bind there because it is competing with the active site. This is known as competitive inhibition. We're binding at the active site. The inhibitor bound at the active site. Okay. We also have a way that we can have our enzyme, normal enzyme, and then our inhibitor binds somewhere else. And when it binds in that somewhere else, this causes our enzyme to change its shape and maybe close its little mouth. And now our substrate can't bind there. This type of inhibition, obviously, we call it non-competitive inhibition. I know you're saying things, Maria, but I'm going to try to ask them before I bring up your statement because I want to see if anybody knows if we're not binding at the active site, we're binding at another site, the other site, but it has a name. In non-competitive inhibition, the site where the inhibitor binds, who do we have left? Kaylee participated. Oh, Dylan, you answered. I guess we've called on everybody then. Yes? Everybody's been called on? Well, I kind of got you answering another question, so I can't, but do you know what this site is called? Easy enough. Okay, regulatory. Yeah, that's fair enough. I saw you participate in other things, so I gave you credit, but regulatory or allosteric. Both of those are interchangeable. Yes.
Medical side.
Mhm.
I know the reason for not is competitive.
It literally binds at that same receptor that like fentanyl would. Yes. I don't know of any like specific examples. I'm sure like if somebody were to be like, tell me about how something works, then I would be like, oh yes, that's non-competitive inhibition. But putting me on the spot like that, that's a good question. I don't know any necessarily off the top of my head, good examples for that. Yeah, they can see your chat when I open it up, Maria. So that's why sometimes I don't open it up because I don't want to give away your answers. But yeah. So all right. Right. So I don't know any like off the top of my head ones, but there definitely are some. And if I were to think of them, I could probably tell you. Yes. So, and that Narcan, the way that Narcan works, to block fentanyl, basically, it's usually fentanyl that we're the most worried about with our narcotics because it can become so toxic at very, very low doses. But there's other, of course, narcotics that can cause a similar issue like that. But instead, Narcan, it will favor binding that, and it's not as toxic to us. Narcan can still cause symptoms and association with narcotic use, but it's less dangerous and toxic for your body. I want to be clear about that too. So it's not non-toxic. It's just better than the actual like, narcotics, the opioids that we're usually combating.
Was that carbon monoxide will Possibly. But like the-
That's what I was gonna bring up next. So carbon monoxide actually favors binding to your hemoglobin compared to oxygen. It outcompetes oxygen in that site, and it binds more tightly even than carbon dioxide does. But you know what works even better than carbon monoxide at blocking that? Cyanide. Cyanide has a very heavy affinity for hemoglobin, more than carbon monoxide, carbon dioxide, or oxygen all combined. It takes very little dosages of cyanide to block the hemoglobin. That's why it's such a potent, poison, basically. Make sense? Similar idea, competitive inhibition. Yeah, just to ensure that nothing else is going to outcompete it. Exactly. Yes. All right. We're tracking on this because there's going to be some questions about this, all of this coming up. We're going to crank through them.
I have a question
Yes.
on the inhibition and the regulation that you just went over. Do we need to note what repression is and induction?
Yes. So oxidation and reduction, we can go over oxidation and reduction. You want me to review that too?
If you want, I was just wondering if the repression and induction is that specifically to the substrate or the competitive inhibition and the non-competitive?
I get what you're saying. So for like regulating the enzymes at like the
Yes.
genetic level, for example. Because we can regulate the enzymes by using inhibition on the enzyme itself, or we can have action on the DNA. So we learned a little bit about controlling the DNA with operons. Remember that? So we can have repressors that literally bind on the DNA and prevent transcription, for example. But that is at the genetic level. So we can control our enzymes at the genetic level with proteins that can block or initiate expression of genes, or we can have regulation of enzymes after the enzyme is made with this type of inhibition. So they are different. Does that sort of get at what you're asking about, Maria?
Yeah, because I guess that repression and induction on what we're talking about, it confuses me with that stuff that we went over on lab where like if you're pregnant you get induced.
That's the
Like
operon stuff.
the
Yeah.
operons, yes. Sorry. Never
Yeah,
mind.
yeah, yeah. So that would have us, I'm of course going to cover it again in my next lab tonight. And of course, you're not going to be there for that, but it's the same kind of idea with it. I will say that controls the expression of the enzymes at the genetic level.
Okay.
This controlling the enzymes after they've been expressed. We can still control them with the inhibitors here. Now, if we're controlling them at the operon level, an inducer is something that can cause a repressor to unblock that gene, which is what we talked about with our inducible operons. Does that make a little
Okay. Yes.
still
Thank you.
that'll regulate expressing the enzyme, but then once the enzyme is expressed, we use competitive or non-competitive inhibition to control whether they're working or not.
Okay, so do we need to even relate that to this
I
right
mean,
now?
the fact that you're thinking through it at this level is going to be more than enough for understanding it for the exam. Because
Okay.
we're tying them both in at the same time in one question. There usually be
Okay.
ideas.
Okay, thank you.
Yeah, absolutely. All right. Okay, so substrate binds to the active site. Good, you guys got that one. We'll move on to the next question. What are causes of a denatured protein? We'll work on erasing this while you guys answer that. I'm going to leave up our different levels of protein structure. This is the slowest erasing that I've ever done in my life. Okay. Proteins can be denatured just like DNA can be denatured. Now we'll go over a little bit about DNA denaturing. We learned about it some in chapter 8, but it's going to directly apply to what we're going to be talking about in lab today, right? But the basic idea is if you use heat or pH, you can disrupt these hydrogen bonds, disulfide bonds, or ionic bonds. in the secondary or tertiary, or even how these things interact with each other with our multiple polypeptides. So you use enough heat or enough extra H pluses. Heat, obviously increasing the temperature. Those weak associations are easy to break with heat. They don't want to hold together if you add enough energy into that. So we'll just degrade down to just being a string of amino acids at that point. But you add enough heat, you incinerate that sucker, then we're going to burn it to ash. And so we know burning something to ash is going to be removing carbon dioxides and all sorts of stuff through incineration. So now we're breaking covalent bonds. So heat can break even covalent bonds there. pH is not going to be as effective at that because pH is just dealing with H plus concentration, but you get enough H pluses in there, and now those H bonds that are holding the structures together won't work either. Or you get too few H pluses. Yes.
So, I have kept aquariums for a while.
Mhm.
My water in Yukon is about the softness relative to possible. So when I have pH crackers sometimes, I think there's stuff like that for that. On pH scale that. And so, when it crashes, if things die because they don't have the hydrogen bonds?
Yep.
Okay.
Yes. Yes. And this is a very delicate balance. So just a little bit. So when I tell you guys our body pH is 7.35 to 7.45, this is a hard boundary. If you're outside of that pH range, you will be sick. And so that's the same thing with any living creature. Some living creatures have a little bit more variability and a little bit more leeway in there. They're not all 7.35 to 7.45, depends on the creature, right? So yes, it's the same idea though. If our pH goes down too much, then we are, messing with those hydrogen bonds, and that's why you get sick. Your proteins start messing up. It's the same thing with fevers. What is fever? Heat. If your body's temperature goes above 105 degrees Fahrenheit, you start denaturing your proteins. And so you start having brain damage, where that's going to start seeing the most effect immediately. And that brain damage can be permanent, and your own body can do that to you. This is why we do not tell people do not take Tylenol. We do not tell people to not take Tylenol, because if you don't take Tylenol, your fever will get worse, and then you're going to have brain damage, either you or the baby that you're carrying. So talk to your doctor and take the Tylenol as prescribed by your doctor. Yes.
Yeah, more at 7.5. And then the options were, you'll get, it's hot, it's higher, but not as concerning.
Mm-hmm.
Chose, no, it's in your outcome, this is, it's the concern option. But I all, I thought about that one because the context with which I tried, with which I tried to answer the question seemed like it would be correct in some telling.
Yeah. So if you want, what we can do in the lab or after lab, either today or next week, we can look at that question, the exact wording of the choices there, and compare it back to this now that we have reference. Okay. Because I don't remember the exact wording of all the choices. So yeah, we can look at that though. Yeah. That's a good question because it relates directly back to this. Now, chapter 10 doesn't talk a ton about denaturing the proteins. It talks a little bit about denaturing the proteins, right? And the same exact principles apply to denaturing your DNA. But when we start getting into unit 3, chapter 11 directly relates back to this, because most of the methods that we use for microbial control use heat or pH or chemicals to denature proteins and DNA. And so having this foundation leads us into unit 3. That's the first chapter in unit 3 that we have. Okay, so we're following with the general concepts of this. Okay. I'm going to go ahead and delete this image. Okay, so I know we're at 6:38, but like I said, we're going to feed a little bit into lab time, and lab should be shorter, and we should still get done early, just FYI. Because I want to get into aerobic respiration and stuff like that really quickly. All right, we already did non-competitive inhibition, but which of these associated with non-competitive inhibition? So we're not competing with the substrate. Where will we be binding if we're not competing with the substrate? So the substrate binds at the active site. So if we're not competing with the substrate, we're not binding at the active site. Instead, we're binding at the allosteric or regulatory site. Those are the same term, interchangeable. Okay. I think the next part we're going to get into. Okay, 5-carbon sugar found in ATP. This is an image of a mitochondria, which is also relevant to what we're about to talk about, right? Here is this. Was it this? That 5-carbon sugar in ATP is ribose. How do we know it's a sugar? Ose. Okay, I'm not drawing the whole sugar. You're welcome. Okay. All right. So it's attached to adenine. Again, you're welcome. This is my adenine. Okay. So it's attached to adenine. It's also attached, if we're talking about ATP, to phosphates, right? PO4 to PO4 to PO4. Remember that PO4 is actually, now, if we're in the middle of all that, then you have another covalent bond to another thing. But one of these is hanging out like this. So all of these guys, negative charge, negative charge, negative charge. So how does ATP hold energy? So I don't mean how did we get ATP? I know that's glycolysis and Krebs and the electron transport chain. I know that. How is ATP holding that energy? From all of those steps, we harness the energy in the form of ATP. This is because these negative guys are repelling each other. And this last negative guy is really repelling each other. It's like south poles of a magnet. They really don't want to be held together. And this bond is a covalent bond. They're being forced. We're forcing oxygens to share electrons there, and they are negative. And they do not want to be covalently bonded. But we forced that through glycolysis, Krebs, and the electron transport chain, all of that work to force that bond to happen, just so that we can break it. And then this releases the energy. Often we're removing that phosphate to another molecule. Often that's what's happening. And if you can imagine, if we're a protein and all of those hydrogen bonds and little interactions and attractions are dictated by those R groups and how they're interacting, and now I'm sticking on a negative phosphate onto them, that's like tacking on like a hot spot on your back. You're going to be like, ooh. The same thing with the protein. It's like, ooh, and it doesn't like that either. You're tacking on that negative thing, it causes the protein to change shape. And now that because that protein changed shape, that's what drives the next reaction going on. Okay, so the phosphate is really what's driving that. It's the phosphate part of the ATP because of the negative charges there. So the whole idea here of breaking this bond, which I know is what you're asking here, what's actually breaking that bond is whenever ATP comes up to, I don't know, DNA polymerase, we're going to tack a phosphate onto DNA polymerase. We're going to make a DP as a result. That causes DNA polymerase to change its shape and then allow us, whenever we see an A, to tack on a matching T on the other side. And then it will reset. And then it'll move on and do it again, and so on and so forth. That's how most enzymes will work. It can be a little bit more complicated than that. If we were getting into biochemistry, then we might have the substrate gets the phosphate on it or something like that, or the coenzyme gets the phosphate. But something dealing with the phosphate moving to something is causing changes of shape that is allowing those proteins to act as the catalysts that they're meant to be. And it can just reset and keep doing that over and over and over again. Okay, I hope that answered that question. So that's broad picture answer to that question. Okay, so that's how ATP works as our energy source. Let's talk a little bit about how we make ATP. Let me clear these drawings. That's good. I want to pick the right color for this because I don't want to get. I'm trying to think what I want. Okay. No, hold on. I want to start over here. We're going to start with aerobic, anaerobic, and fermentation. These are all different types of respiration. Yes, I know you think respirating is to breathe. Why do you think you breathe? This is the only reason why you breathe, is this. And I mean that literally, okay? So why do we inhale oxygen and exhale carbon dioxide? This is the only reason, is this. So I cannot emphasize it more, how important this system is for existing in life, all right? Aerobic respiration has 3 steps. Glycolysis, no. Krebs, and the electron transport chain. The electron transport chain, last step, our last electron acceptor in aerobic respiration is oxygen, right? Aero, air. If you are anaero, you are not oxygen. So it is quite the same. Glycolysis, Krebs, and an electron transport chain. The only difference is you are not oxygen. That's it. That's all we need to know for now, okay? What else could it be? Sulfate, nitrite, nitrate, all sorts of stuff. We're not going to worry about what. You can worry about that at lab. Our very last lab, we learn more about this, okay? So we'll wait. That's at the end of the semester. But for now, not oxygen is enough, all right? If you ferment, this is what some organisms do when oxygen isn't present. Even your own muscles can ferment. And we'll talk about how fermentation fits in with this as we go through all of these steps, okay? All right. The first step in aerobic respiration is glycolysis. In glycolysis, we are taking a 6-carbon glucose, which is C6H12O6, and we are turning it into 2 3-carbon pyruvic acids or pyruvate. Now, in the body, pyruvic acid and pyruvate are essentially interchangeable terms. I don't care which one is which or why or how. They're essentially interchangeable terms. When we do this, we happen to make a net 2 ATP. And we happen to make some NADH. NADH, for our purposes, represents this H, means that we are carrying 1 electron with us. So this serves as essentially and effectively an electron. The goal, I will write in orange, the goal of glycolysis is to make pyruvate in aerobic respiration. That's our goal. You're welcome. I know. We didn't go through glucose 6-phosphate and all this other stuff, did we? You're welcome. Okay. Right, because that's not as important to me. How many of you guys got lost in that in chemistry and stuff? Yeah? It's really easy. And I don't know why they focus on the steps, because this is way more important. I care way more that you know the whole point of glycolysis is to make pyruvate. And why is pyruvate important? Of course, the next steps will tell us why. Okay. But for now, if you are going through fermentation, I told you all you do is go through glycolysis. So in red, we will put fermentation, okay? If you ferment, your goal is to make ATP. How do we make that ATP? It's right here, right? But that's the only source of ATP. We got 2 ATP out of glycolysis. That's not the goal of glycolysis if you're going through aerobic respiration, but it is the goal of glycolysis if you're just going through fermentation. The problem with fermentation is, what do we do with this electron? You can't just have electrons collecting up. So what it does is they feed it back in to pyruvate. Now it's not that simple, because this is chemically changing pyruvate. So when we chemically change pyruvate, it then becomes lactic acid or ethanol and or carbon dioxide. In fermentation, we got to get rid of that electron, and that changes pyruvate to those things. There's other things that can change it to too, but those are the main things. So this is why when we ferment beer, we get alcohol. This is why whenever you ferment yogurt, you get lactic acid. Why it has that sour taste. We're tracking that? This is why whenever you run a lot and you end up with sore muscles, when we say you have lactic acid buildup, it's because you ran out of oxygen getting fed to those organs, your muscles. You didn't get enough oxygen to them, so it had to ferment in order to make enough ATP just to stay alive. So it fermented. All right, we tracking that? It's good. All right, so that's where fermentation falls in. It's in the red. All right, next thing. Next step. That was it. That was glycolysis. Like I said, you're welcome. Okay, next step is that intermediate step. In the intermediate step, we are taking that lovely pyruvate and turning it into acetyl coenzyme A. Pyruvate is 3 carbons. Acetyl-CoA is 2 carbons. What happened to one of those carbons? It came off as our first exhale, carbon dioxide. This is why you exhale carbon dioxide, step 1. There's, of course, more carbon dioxide in the next step. Okay? So we exhale some carbon dioxide. We also make a little bit of NADH. And remember, NADH is effectively an electron. The whole purpose of that intermediate step is to make acetyl-CoA. The next step is the Krebs cycle, also known as the citric acid cycle. Okay? In the Krebs cycle, we're going to take that 2 carbon acetyl-CoA, combine it with a 4 carbon. It happens to be oxaloacetic acid or oxaloacetate, but I'm not going to test you on that. Again, you're welcome. A 4 carbon thing plus a 2 carbon thing makes how many carbons? 6. They're additive. That happens to be citrate. That's why it's also called the citric acid cycle. Remember, citrate and citric acid, same thing. And it's going to cycle through some stuff. How do you get from 6 to 4 carbons? You got to kick off some carbons. So they do. We have a couple of instances of carbon dioxide getting kicked off. That's waste. Okay, yeah, exhale it. So we're exhaling carbon dioxide here, here, and here. What's also made while we're kicking off those carbon dioxides is going to be NADH. And it doesn't matter where you are putting these in your cycle. I don't need you to know all the steps and where exactly these electrons are going and coming from and everything. Again, you're welcome. I want the big picture idea. These guys are our electron coenzyme carriers. Each of these guys represents electrons. As it turns out, and we make a little ATP here too. I forget. Cool. So we do make 1 ATP per glucose, each pyruvate. So 2 per glucose. Each pyruvate, we get an ATP. How many pyruvates do we get from glucose? 2, right? All right, so are we following the idea of what's happening with this? What's the purpose of the Krebs cycle? The goal? To make electrons in the form of FADH2 and NADH. So now, can we please summarize this? Glycolysis, we made pyruvate. Intermediate step, we turned pyruvate into acetyl-CoA. The whole reason we needed acetyl-CoA is to break off some electrons. Why do we need electrons? That's the big daddy, right? That's the electron transport chain. Are we following? Hope so. Because the next last part, obviously, we are going to be taking these electrons here into the electron transport chain, which I'll summarize up here. The electron transport chain or the electron transport system on the exam, and you'll hear me talking about it all the time. I learned it as the electron transport chain, the ETC, okay? The ETC, what is it? The whole purpose of the ETC, the goal, is to take electrons and turn them into ATP, make ATP. So the whole reason we did glycolysis was to get pyruvate so that we could turn pyruvate into acetyl-CoA, so we could steal electrons from it and shuttle them through the electron transport chain. So here's how it works. We'll have a membrane. On that membrane, we'll have these proteins. These proteins are called cytochromes. Oh, I need to draw this lower. Hold on. I can't just, okay. That works. Here's our cytochromes. That works. Then we have this big old blobby guy here. This is ATP synthase. Okay, and we got more membrane after that. In bacteria, we have our cell. I have a student that's supposed to be coming to our lab tonight, and I just remembered that she would be meeting me in the lab right now. Let me check that email real quick. All right, so we have our bacteria. This is your inner membrane. We also know this as the cytoplasmic membrane. This is where your electron transport chain will be. In eukaryotes, you have a big old cell. Here's your nucleus. Here's your mitochondria. No, this is not to scale. Okay, we'll survive it. This is the inner membrane of your mitochondria. One single mitochondrion. The inner membrane of the mitochondria is also known as the cristae. So all of this is happening on that inner membrane. But do you see how they're really the same place? Because remember that in our eukaryotic cells, mitochondria used to be bacteria back in the olden days. We have a lot of evidence that supports that. So it's really the same place. Okay, so that's where our electron transport chain is and how they differ between each other. You can open my email, it's totally fine. You can totally do it. You don't have to fight me on this. Thank you. Okay, so, all right, so NADH, you can, yes. I'm sorry, if you want to, so I know that we have students from the other class coming and joining. We're going over lecture a little bit. So we're going to be like, I want to say another probably 15 minutes.
Okay, I'll just tell them.
You can come in here and hang out if you want to. Yeah. We're running a little over. Okay. Yeah, lab should be shorter. So we're taking a little bit more lecture time. So yeah. Okay. Anyways, NADH and FADH2. These H's are going to become electrons. And so they're going to, if you are an H that is covalently bounded to something, you are sharing, here's whatever you're covalently bounded to. You are sharing electrons, right? You have an electron and the thing you're bound to has an electron. If I take the H off, then you will become H pluses and the electron that you had with you. That's what's happening here, okay? So these H come off, their electron separates from their H pluses. We tracking that a little bit? Okay, so that's what's actually happening there. The electrons themselves, thank you, Asley. The electrons themselves are going to shuttle through these cytochromes. Again, we got a negative electron moving through things that are held together by attraction forces. That's going to cause them to change their shape. And while they're changing shape, they're going to grab onto these protons and shoot them out. And we're going to collect a ton of protons out here. This is going to create a gradient. This proton gradient is also called the proton motive force. It's a force now. We created that gradient. It wants to equal out on both sides of the membrane. So some of those protons come back in. We can't let them build up though, right? If they build up, then it's equal on, it's iso on the two sides. And now they don't want to keep coming back in. That's bad because when they come back in, what happens is we take a DP and turn it into a TP. This is the goal of the whole thing by adding on an inorganic phosphate. The movement of those H pluses, again, if the negative caused something to change shape, just like pH causes them to change shape and interrupts their bonds, moving a proton through changes their shape and interrupts their bonds. When ATP synthase sees that proton, it goes, grabs that thing, sticks it onto this other thing. And that's the change that's happening from that proton going through. So ATP synthase drives creating ATP because that positive proton is moving through it. And it will just keep doing that over and over and over again because it's the only way the protons can come in. So we can't have buildup of the protons. We can't have that. We need to get rid of them. We also can't have buildup of the electrons here. We need to get rid of them so we can keep this cycle going over and over and over again. So we will take, let's see what color I wanted to do. Let's do red. I don't think red was the right choice, but whatever. The proton and the electron, now we've got just regular hydrogens. They can share with other atoms and half of an O2. What's half of an O2? Just an O. And now we can have oxygen sharing electrons with hydrogens. And what's this molecule? Water. This is why when you are stuck out in the desert and you aren't drinking any water, you still continue to pee because you still continue to make water as a byproduct of the electron transport chain. Now, your body, your mitochondria, this is all mitochondria do. They do nothing else. Aerobic respiration. All mitochondria, aerobic respiration. Plants are eukaryotes. All eukaryotes have mitochondria. Plants use sunlight energy to fix carbon dioxide into glucose so they can put it through aerobic respiration. Aerobic respiration. So plants also use oxygen. They don't just make oxygen. You make more than they use. Okay? They have mitochondria. They also do this. Are we tracking? Okay. All right. This is why you breathe in oxygen. This is the number one only reason why you breathe in oxygen right there. To keep that gradient stable. To get rid of the extra protons and the extra electrons. So we can keep the electron transport chain pumping out ATP. So much so that if I suffocate you right now, you die in minutes. This is the only reason why you breathe in oxygen. It is the only reason why you exhale carbon dioxide. But we can see here that this oxygen that you use that makes water, it doesn't make carbon dioxide. The sugar, food, or related items that you ate that can feed in at any of these points made carbon dioxide. You breathe in oxygen because we need it for the electron transport chain. You exhale carbon dioxide because we used sugars in glycolysis, the intermediate, and the Krebs cycle. They didn't touch each other though, other than mixing in your lungs. They didn't chemically interact. Is that not cool? Tell me this isn't cool when you see the big picture stuff. You literally only breathe in oxygen to keep that proton motive force going. That is the only reason why you breathe. That is so cool. The only reason we breathe in and out is this. That is it. So much depends on it. Okay. Same for bacteria too. Any energy that they need. Now, if oxygen is your final electron acceptor, you're aerobic. If you use something else as your final electron acceptor, then you're anaerobic. If you're fermenting, then you stay stuck in the red with glycolysis. All right, any questions about that? You notice how I didn't make you know glucose 6-phosphate. I didn't make you know oxaloacetate or any of that stuff because I don't care about it. I want you to get the big picture stuff. What's actually going on in each of these steps? Okay, any questions about glycolysis, intermediate, Krebs, electron transport chain, aerobic respiration? Any questions about that? Do you have any specific questions about photosynthesis? I'm not going to cover it here. Okay, it's in the lecture. I'm just not going to review it here. All right. Okay. Our last thing then is our activity. Again, I'm just going to briefly just summarize it for us. For chapter 10. Wow. Okay. I'm going to try. Okay. DNA polymerase. I guess I'll remove all this stuff. It's just so nice. I did such a good job with my drawings. DNA polymerase is an enzyme that requires magnesium in order to function. Magnesium is a cofactor. Okay? That's the first question. Second question, in the Krebs cycle, acetyl coenzyme A is combined with oxaloacetate to make citric acid, 6 carbons. Then we proceed to transfer electrons from all of those intermediates to make NAD into NADH or FAD into FADH2. And each of those guys, the H's aren't just H's, right? They're coming along with electrons. So this H and these H's are all carrying with them electrons. So we gained electrons. Gain electrons is reduced. Right? Leo says GER or oil rig, whichever one you prefer to remember what oxidation and reduction on. I learned Leo says GER. So did Dr. Tim. I don't know what you learned. I think Dr. Singh, does Dr. Singh do oil rig? I don't know. Whatever works for you, okay? Leo, if you don't know what I'm talking about, then you really need to watch the lecture. No offense. Loss of an electron, loss of electron is oxidized. Gain of an electron is reduced. Oil rig, oxidation is loss. Reduction is gain. Whichever one works for you. I learned Leo says grr. Leo the lion says grr. That's how I learned it, but whatever works for you, okay? Question 3, explain why it's critical to maintain more protons out there. If it equalizes, we stop making ATP. Those protons moving in is what drives ATP synthesis. Question 4, compare and contrast the locations of the electron transport chains of eukaryotes versus bacteria. In bacteria, it's on their inner cytoplasmic membrane. For eukaryotes, it's not on the cytoplasmic membrane, it's on the mitochondrial inner membrane. It's actually where we lose ATP because glycolysis, Krebs, and the intermediate steps all happen in the cytoplasm. In bacteria, that's right next to their membrane. In our cells, we have to move the electron transporters, NAD and FAD, into the mitochondria to shuttle them through the inner membrane of the mitochondria. And that requires energy to move them. So we lose ATP there. That's why bacteria make 38 ATP and eukaryotes make 36 ATP. 34 of our ATPs are made in the electron transport chain. Okay, it's quite a lot. Those fermenters really missing out on some ATP. Why does fermentation generate, does fermentation generate more, less, or an equal amount of ATP compared to aerobic respiration? Like I said, in fermentation, you're making 2 ATP as opposed to 38 possible ATPs if we are going through proper aerobic respiration. Okay? Okay. All right, that's it. So we can head up to the lab and I'll see you guys up there. Try to head right up there if you can. If you need a couple minutes, take a couple minutes. I'll meet you guys up there. Who's heading up right to the lab right now?
I will.
Whoever can take my key and unlock the lab. Oh my gosh, it's so close to my face.